Imagine spending 2 years developing a Leuprolide depot generic. Your formulation looks perfect, analytics match, and then your bioequivalence study fails just because your microsphere particle size shifted slightly during scale-up. That's the reality of long-acting peptide depot development. Hi, I'm Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories, Canada. At Resolve Mass, we specialize in solving most complex challenges in drug development, from peptide characterization to impurity identification, supporting your journey from discovery to regulatory success. When companies start developing a Leuprolide depot generic, they often assume it's just another injectable. But very quickly they realize they are not dealing with a simple solution. They are dealing with a polymer controlled drug delivery system. You are essentially designing a microsphere engineering project, not just a formulation. The peptide is encapsulated inside PLGA microsphere, and after injection that polymer slowly degrades, releasing the drug over 1 month, 3 months, sometimes even longer. Now here is the challenge. Every single variable matters. Change the polymer molecular weight, release changes. Change the solvent system, morphology changes. Change mixing speed, particle size shifts, and suddenly your release curve no longer matches the reference product. This is exactly why choosing the right CDMO becomes a strategic decision, not just an outsourcing decision. Now let's talk about real industry challenge. We have seen programs where the lab scale formulation looks identical to the reference product, particle size matched, encapsulation looks good, but during scale up, emulsification dynamics change slightly, and that caused wider particle size distribution, and the result, the early burst release increased and the bioequivalence study failed eventually. This happens more often than people think because in depot formulation, microsphere properties directly control clinical performance, not just formulation composition. So the first thing I always look for in a CDMO is real PLGA microsphere experience, not just peptide experience, not just injectables, but actual controlled release microsphere development. They should understand double emulsion solvent evaporation, polymer degradation kinetics, drug polymer compatibility, encapsulation efficiency optimization, residual solvent control, because these are not theoretical. They determine whether your product releases the drug correctly or not. Where analytical capability becomes critical. The next major differentiator is analytical depth. For standard generics, you might rely heavily on assay and impurities, but for Leuprolide depot, that's nowhere near enough. You need to understand particle size distribution, polymer molecular weight changes, microsphere morphology, peptide degradation inside the polymer, long-term in vitro release profile. This is where many programs struggle. Release testing itself becomes a development challenge. We have worked on cases where release testing variability was so high, teams couldn't even compare batches confidently. At Resolve Mass, one of the approaches we use is multi-time point orthogonal characterization, combining release profiling with polymer degradation monitoring and peptide impurity tracking. This helps identify whether variability is coming from the polymer erosion, diffusion behavior, or peptide instability. That level of insight can save months of troubleshooting. Another major risk is manufacturing scale-up. A lot of CDMOs can overlap microspheres at lab scale. Far fewer can scale them reproducibility. And what is the reason? Because scale changes mixing hydrodynamics, solvent evaporation rate, shear forces, emulsion stability, and all of this influence microsphere formation. So when evaluating a CDMO, ask, have they successfully scaled PLGA microspheres before? Do they run pilot scale batches? Can they control particle size during scale up? Because lab success doesn't guarantee commercial success. Sterile manufacturing often underestimated. Leuprolide depot is also a sterile injectable, which adds another layer of complexity. You need sterile microsphere handling, lyophilization optimization, powder filling precision, vial or syringe compatibility, and importantly, microsphere integrity must remain intact during fill finish. We have seen cases where the formulation was perfect, but lyophilization cycle caused aggregation, altering release. That's why CDMO infrastructure matters as much as formulation expertise. The most difficult part, and that is in vitro release testing. If there is one area that determines success, it's release testing. These studies run for weeks and months. They must stimulate physiological condition, and they must be reproducible. Without reliable release testing, you can't optimize formulation, you can't compare to reference, you can't build IVIVC, and you can't support knee study. In complex programs, we often design accelerated release models alongside long-term studies, so development decisions don't take months. That dramatically speeds up the optimization. Now let's talk about peptide stability. That's the hidden risk. This is one more complication actually. Leuprolide is a peptide, and peptides are sensitive to oxidation, hydrolysis, aggregation, temperature, and solvents. And during microsphere encapsulation, all of this risk increase. A CDMO must understand peptide degradation inside polymer matrix, not just solution stability. This is where combined peptide analytics plus polymer science becomes essential. Finally, let's talk about regulatory strategy for complex generics. Regulatory expectations for depot generics are significantly higher. You need comparative characterization, release similarity, structural similarity, B strategy alignment, ANDA documentation. Regulators often expect multiple layers of evidence, not just bioequivalence. So CDMO regulatory experience becomes a major advantage. So when evaluating a CDMO for, let's say, Leuprolide depot, I recommend checking, do they have PLGA microsphere experience? Do they run long-term release studies? Can they scale manufacturing? Do they handle sterile fill finish? Do they understand peptide stability? Do they support ANDA strategy? If any of these are missing, development risk increases significantly. And Leuprolide depot generics are not standard injectables. They are polymer controlled peptide delivery systems, and success depends on many parameters, many variables. For example, microsphere formulation expertise, advanced analytical characterization, scalable manufacturing, reliable release testing, peptide stability control, regulatory strategy. Choosing the right CDMO early can save years of development time for the client. And if you are working on a Leuprolide depot or any other long-acting injectable generic, the most important decision isn't just formulation design, it's partner selection. Because in depot development, small differences in expertise create big differences in outcomes. Thank you once again for watching this session. and see you in the next one.